All right. Uh, Great to see you all. Welcome everybody to the VMOL seminar. It's my great pleasure to have Trent Northen today here with us. Yeah, Trent is one of the experts in non-targeted metabolomics, I would say, and fellow DOM enthusiast. So yeah, it's really fun to have you here, Trent. Just to give you all a very brief background about Trent's bio, he did his bachelor's in chemical engineering at the University of California in Santa Barbara, followed then by a PhD in biochemistry at Arizona State University, before he then joined the SUSTAC lab as a postdoc at the Scripps Research Institute in San Diego, and then followed by now being a senior staff scientist at Berkeley, where he runs the Northen lab and also a lot of collaborative metabolomics work. So I'm sure many of you know him. And yeah, without further ado, floor is yours, Trent. Looking forward to your presentation.
Thank you so much, Daniel and Allegra and Ming, for inviting me to speak today. It's really fun to get to talk to an international crowd. So thank you all for joining. I'm at Berkeley Lab, which is located basically up the hill, as we say, from UC Berkeley. And so this is one of our big landmarks, which is the cyclotron. And I'll say a little bit more about that in a second. But you can see the Golden Gate Bridge down here in San Francisco. So a nice place to visit if you're ever in town. I'm also at the Joint Genome Institute, which is a user facility. So today I'm going to be talking about the rhizosphere, which is the region of soil immediately around plant roots. And it's a very interesting environment and also very important because a large fraction of carbon on Earth is cycled through the rhizosphere. So plants deposit up to about 30% of their photosynthate into the rhizosphere, where it drives and feeds beneficial microbes that help with the plant growth. But it's often very challenging for companies or for technologies to get beneficial microbes to grow in the rhizosphere. Just very much like the gut, where you maybe take probiotics and they don't stay. You have to keep taking them over and over again. So there's the same problem in the rhizosphere. And understanding these processes are really critical to sustainable agriculture. And so, yes, I'm a chemist. So when I look at the rhizosphere, I imagine all these little microbes. And then, if you really want to understand what's going on, what's critical is the chemistry, because this is how the dissolved organic matter is processed, how the plant is interacting with the microbes. It's through these small molecules. And, that's definitely a chemist's perspective of the world. But my overarching hypothesis is that if we can understand the chemistry, we can understand a lot of the processes, critical processes, and including ecological processes in these environments. So it's been known for a very long time that plants exude a diversity of metabolites. So they're basically, like I told you, fixing carbon, pumping it out through the roots, where it performs all kinds of functions like mobilizing nutrients that are absorbed onto mineral surfaces, interacting and feeding microbes. You have nod factors that recruit specific nitrogen-fixing bacteria, so all kinds of factors. And because so much carbon is going into the environment, and because this is benefiting the plant by selecting for beneficial microbes, it's really critical for improving productivity and resilience. And because it's so much carbon, it's also really important to understand Earth's carbon cycle. And so this is why my lab is focused on the rhizosphere. So I'm gonna basically cover both of these today briefly. So I'll talk a lot about plant microbe interactions. And then I'll also tell you about some of our latest research, including with Daniel and Ming, on some new tools for studying what's happening in soils at the molecular level. So there's many countries and a lot of reports out there that saying if we want to achieve sustainable agriculture, we need to learn how to harness beneficial microbes. Plants evolved in the context of microbes. Microbes were here first, so they've naturally evolved to benefit from microbes that do all kinds of different functions for them. However, because of the way we've managed lands, we've lost a lot of these beneficial microbes and their interactions. And we can also engineer bacteria that have new functions like bacteria that will grow in the rhizosphere of maize and fix nitrogen for that. And so this was recognized as one of the best inventions in 2023, this technology from Pivot Bio. But as I mentioned at the beginning, one of the really big problems is that these work sometimes and they don't work other times. And so it's a big risk for farmers because it's expensive to have these and you apply them and you never know if it's going to work or not. And that's because it's a black box. We don't really know how these work. And this is also true in the gut, right? And so these companies here, make it a routine, basically take it every day, right? So maybe you could do this with a probiotic. You could take one every day. But a farmer cannot go apply a probiotic every day in a field, right? That's just impossible and would be way too expensive. So we need to figure out how to get this to work one time. And so what we've been working on for a long time now is trying to understand how the exudates select for beneficial microbes and which metabolites in particular are important for selecting microbes. And so it's been known for over 100 years that when the presence of a root in soil changes the microbial community, and this is known as the rhizosphere effect. And you can see it here. So these actinobacteria, they start off as being abundant, and this is each of these bars is a different time point. And as the plant's growing, the actinobacteria become less abundant, and these alpha proteobacteria are becoming more abundant, right? And so we hypothesize that root exudates are explain these differences. And so we collected root exudates at all of these different time points. And the first thing we wondered about is, does a plant change its exudates over time? And the answer is, at least for this one, yes. Early on, the plant is releasing mostly sucrose and homoserine. As it matures, it produces a wide array of molecules, including many aromatic compounds. And then later, as the plant is getting old, it produces these senescence hormones. So what we did is we took these exudates and we fed them to bacteria using exometabolomics, which is just simply where you compare inoculated and uninoculated media to identify what's being used by the bacteria. And we found that the bacteria that become more abundant in the rhizosphere prefer to use these aromatic compounds, and they're not preferred substrates by the bacteria that become less abundant in the rhizosphere. So this is sort of our entryway into this idea of sort of matching plants and microbes using metabolites. So we next hypothesized that if these metabolites are really being used by the plant to select for beneficial microbes, they should become more abundant when the plant is in need of help. This is the cry for help hypothesis, that when the plant is stressed for nutrients, these compounds should become more abundant because the plant needs to recruit more of these microbes. So we designed this huge experiment. This was led by Mary Firestone, actually required a whole truckload of soil. Any experiment you can do that has a big dump truck, that's a very cool experiment. And so we packed these giant columns with soil from the field, and we compared the metabolites and the microbes in the rhizosphere under different nitrogen stress conditions. So this soil that we used is a marginal soil, so it basically doesn't have any nutrients in it or very, very low. And then we added fertilizer. And the idea is that when we add lots of fertilizer, metabolites that are needed to recruit microbes that are able to fix nitrogen or mobilize phosphorus will become less abundant than in the marginal soil that we start off. And consistent with our hypothesis, we found that under nitrogen stress, metabolites, many of these aromatic metabolites become more abundant. And we could do a correlation analysis with the microbial community composition to identify what we were calling hub metabolites. So these are the metabolites that explain most of the positive and negative correlations between the community structure and the microbial community composition. And so we also call these keystone metabolites. So the metabolites are these little boxes, and these circles are the microbes. And we can see that these aromatic compounds had the most both positive and negative interactions. So they're selecting against some microbes, and they're selecting for other microbes. If you have a sharp eye, you will notice this very interesting metabolite here, serotonin, which is a metabolite that is associated with feeling good in humans. And there's a ton of research out there on how that gut microbes trigger the human host to produce more or less serotonin. So here we found it in plants, and that was pretty neat. It turns out that there is some literature precedent on this, but we decided we had to do this fun Friday afternoon experiment where we took plants and we gave them serotonin to see if it would make them happy too. And what we found is that when we added serotonin at 100 micromolar, we got plants that were almost twice as big. So serotonin made for some very happy plants, and we thought this was really awesome. You can see how big the plants were when they had the serotonin. So this is an example of a metabolite that if you have an interaction with a microbe that triggers more serotonin production by the plant, you could get a bigger plant. So that was really fun. I also mentioned that the serotonin is one of these hub metabolites, so it promotes the growth of some microbes and not others, or at least there were positive and negative correlations. So we had some happy microbes and some unhappy microbes. So we made isolates of these bacteria, and we tested if we added serotonin, do the ones that were positively correlated become more abundant, and did the microbes that became that were anti-correlated become less abundant. And you can see here, this is increasing serotonin concentration. The ones that were positively correlated in general, sort of directionally, one of them was significant, grew better. And the microbes that were negatively correlated with the abundance of serotonin at high concentrations were all inhibited. So this is some suggestion that these molecules can play this sort of dual role in the rhizosphere. So this is all nice, but it's correlative. And so we wanted to get towards causation. And I showed you that cyclotron on the hill, big facility. And at Berkeley Lab, we like to say that we're the home of team science, where you bring together these theoretical, so here's Oppenheimer and here's E.O. Lawrence as just kind of prototypical theorists and experimentalists. And so bringing groups of scientists together to work and do things that aren't possible for individual investigators. And so this is a really great model, I think, for microbiome research, because I would say studying microbiomes is just as complicated as studying subatomic particles, et cetera, except we generally don't use a team approach. And one of the reasons why we don't use a team approach is because there's a lack of standardization, and it's almost impossible to reproduce each other's work. And so what ends up happening is that, say, one of you wants to get into studying rhizosphere microbes, you pretty much have to start from scratch. There's not much in the way of resources. And so you can't also read a paper and then replicate it and build on it. And this has been recognized notably by this commentary from the chief editor of the Journal of Bacteriology, which says, I propose that we as a field need to consider consolidating around model communities that will serve the same functions as model microbes. And this is something that we've been working on for a long time. We had a summit called the EcoFab. EcoFab stands for Fabricated Ecosystems. We had a whole summit on it in 2017, and we came up with this vision for how to standardize microbiomes. And so you need to create a reproducible habitat that everybody can get their hands on. You need to create a defined microbiome. You need to have all the protocols for doing the omics analysis. Those all need to be standardized. You need to be able to make spatial temporal measurements because in ecology, that's essential. You would never go take a forest or a jungle ecosystem and first, grind it up in a blender and then try to interpret the metagenome and metabolome, right? You wouldn't do that. You'd go look and say, oh yeah, the monkeys are associated with trees. Like that's really important information. And then you need to do some intra-comparison studies across laboratories to see if all of this stuff is reproducible. Once you have this, then you can start really taking great advantage of machine learning because all your data is standardized, reproducible across labs. And then you can finally get to these really mechanistic type studies. We decided to build our EcoFab using this model grass because we have thousands of mutants of this grass. It's very small and it's relevant to all grain crops like rice, wheat, corn, so major agricultural crops, as well as all the bioenergy crops. So we built this little device, which we call the EcoFab 2.0. It has ports for collecting exudates and metabolites, adding microbes. It's autoclavable, sterile system. It goes on top of a microscope, so you can image where the microbes are. You can also connect lure ports up here for doing gas analysis. So just a nice functional tool. These are all freely available through the JGI user program if you're interested. So next we had to build our model microbiome. So Kate Zalnina isolated 1,000 bacteria from one plant in the field. She also did, we had done a field study. Mary Firestone had led a field study at the same site. And so Kate was able to look at the field results across the literature and design a model microbiome that captured the diversity and abundance of organisms that are commonly found in the rhizosphere. These have all been deposited in DSMZ, and they're now available as a collection. So you can get these very easily, very inexpensively, thanks to them. Karsten Zangler's lab worked out protocols for how you resuscitate them, how you make the microbiome. So this should make it very easy for people to use this community. So now, we've kind of checked all of these boxes. And so now we have the dreaded intra-laboratory comparison study. This was I knew this was going to be hard. This was much harder than we ever dreamed. This was extremely difficult. But fortunately, we had sort of the dream team for organizing this. We had many of the leading labs in plant microbiome research across the world. And so Australia, 2 labs in Germany, 2 different labs in the United States. And we all performed this same study, which is where we're comparing 2 different microbiomes. One is this complete community, so it's a 17-member community. The other one, the only difference is we're leaving out this Burkholderia. And I'll tell you in a second why that's interesting. All of the laboratories followed the same protocol, and we've published the protocol as a protocols.io, so everybody can find it, where they basically germinate the seedlings, they test for sterility, they inoculate the seedlings with the microbiome. After 1 week, they start taking root scans and watering the plant, changing the water media into the plants. And then at 2 weeks, they image them again, change the media. And then at day 22, they sample for sterility and collect the final images. And then all of the labs performed phenomics. So they scanned the roots to look at the morphology of the roots. And then they collected samples for 16S sequencing and metabolomics. Since it's known, particularly metabolomics varies lab to lab, we decided that our lab would do all the metabolomics and sequencing just to keep it nice and simple. So why Burkholderia? Why is this interesting? Well, it's well known that Burkholderia are dominant colonizers in the field. This is one of the really dominant organisms. And if you put Burkholderia in an EcoFab, so here we're looking at a fluorescent Burkholderia, looking from the bottom, one of these EcoFab devices. At the root, it is just coated with Burkholderia. So Burkholderia colonizes the plant like absolute crazy. And so if you do 16S sequencing of the community when Burkholderia is present, it's mostly Burkholderia. So it really dominates in the rhizosphere. And the plant doesn't like it. So you can see here that when you've got a complete community versus the oxygenate, you get a smaller plant. So unlike serotonin, which makes happy plants, too much Burkholderia makes for sad plants. So we've got, big changes in the community structure, phenotypic effects of the plants. So this seemed like this, and it's just sort of a simple experiment. So we thought this would make for a good ring trial. So Vlastivil Novak is the hero of this story. He is the one that made all the protocols, made all of these little kits to send off to all of the different labs. We came up with some data loggers that people could put in their incubators to make sure that they had the right temperature and lights and everything. And actually, we did find that even though we tried to make everything the same, the lighting in the growth chambers was different. So that's one of our big experimental variables. He also made annotated videos for all of this. So the idea, again, we want people to be able to replicate this, so you can watch the videos, follow along, and do all of the experiments. So how did it work after all this effort? Well, the first really good piece of news, and this is the first news that we got, is that there was very little contamination. And of course, this could be a complete, this could destroy the entire experiment if there was contamination. So there were only 2 events across all 5 laboratories where we saw any contamination. And one of those was on a cracked plate. So likely the contamination from the cracked plate. So really, maybe only 1 contamination event across all of the experiments. So that was great. So Blastomil had seen this significant decrease in the shoot weight when Burkholderia was present. How about if you look across all of the laboratories? Across all laboratories, they saw it. You see the same effect, that there's a significant decrease in the plant size when Burkholderia is present. So that was very reassuring. So then we also collected all of these images. So every lab used basically a photocopier to scan the roots. And then Danny Ushizima had written some AI-based, machine learning-based imaging software. And even though everybody was using a different scanner, her software was able to, her software is called RizoNet, was able to extract basically all of the pixels that are associated with roots. And then we could plot it out across the labs to see if we're seeing consistent growth. And this is lab A, B, C, D, and E. And, first of all, the error bars are pretty small. So for plants, this is really good. I mean, plants are gigantic. They're very difficult to grow reproducibly. And we saw consistent decreases in the root length, that was assessed by the pixel ratio, with the 17-member community versus the oxenic across all of them. So that was really nice to see. And how about the community structure? Well, here we see very clear separation based on the community structure and not by the lab groups. Of course, the nightmare would be that it clusters by research groups, not by treatments. And here we get a nice separation that's by that it's 16 versus the 17-member community. Nice clear separation. I mean, there is a little bit, maybe a little bit of separation here by labs, but nothing really dramatic. And then this is looking at the 16S data. So this is the abundance of different organisms in this bar chart, stacked bar chart. And so when Burkholderia is present across all of the laboratories, you see that it dominates, right? And this is the inoculum. So we're getting this really strong rhizosphere effect where you're getting dramatic changes in the community. And when Burkholderia is not present, you see more of this Rhodococcus as being the dominant organism. What about the metabolites? So this is a hierarchical clustering of metabolites on rows, and the columns are samples. This is, unsupervised, it's hierarchical clustering, and the samples clustered by oxenic, 17-member community, and 16-member community, which is just, that's exactly what we were hoping for. And you can see that, the 17-member community, when Burkholderia is present, we get increases in things like thiamine, guanosine, and cytidine. And when it is present, you see less of some of these other metabolites like guanosine and melantin. So it's a nice separation by which organisms. Spent too much time going on this. And so we now have a reproducible system that everybody across the community can use to do mechanistic studies. And so now I want to go through some driving mechanisms, like how we can actually use this. So Blastomil did recently published a paper using the EcoFab to study the effects of plant nutrition on exudation. And one of the most interesting things that we found is that, first of all, our plant really likes ammonium nitrate. So you get the biggest plant when you have ammonium nitrate present. And so we did do sort of a global untargeted analysis of this And you could find some sort of obvious things, like when the plant is starved for nitrogen, there's fewer amino acids in the exudates, but we probably all would have predicted that. But what was really neat is that when the plant is grown on ammonium nitrate, dopamine is very abundant. And this is the first report of dopamine being present in exudates. And look how dramatic this is. So happy plants, you get this dopamine production. And so we thought, wow, this is really neat, dopamine. So we've got serotonin. And this is, of course, another one of these. This is a catecholamine, which is also this is a class of neurotransmitters. And there's many ones that you've probably heard of, like epinephrine, adrenaline, norepinephrine, dopamine, L-DOPA. These are all really interesting compounds. And dopamine is very abundant in the roots. So we did a quantitative analysis, and it's almost 3 milligrams per gram of fresh weight. So tons of dopamine in our plant roots. So and so then we did the same experiment that I told you about with the serotonin, where we added dopamine to see if we would get a happy plant. And dopamine has kind of a concentration dependent effect. So initially you get a significant increase. This is the root weight. But then if you give it too much dopamine, it's not good for the plants. I don't know if this is true with humans, but clearly there's some sort of optimal concentration of dopamine. So we wanted to see, basically do the same sort of correlation analysis with our community structure. So taking our SynCom and identifying which microbes are most correlated with dopamine, and see if it's sort of one of these hub-like or keystone metabolites like serotonin is. So we took advantage of this Brachypodium collection, where John has a whole collection of natural accessions. So these are Brachypodium plants that are natural, but they're just collected from different regions of the earth. Turkey, Spain, just around the Mediterranean, where this plant is commonly found. And so we grew the plants with and without the SynCom. We measured the phenotypes, did metabolomics and 16S sequencing. We then wanted to correlate the metabolites and microbes and see if dopamine is one of these hub-like molecules. We used 10 different lines, and we used the SynCom that I told you. And so this is the Spearman correlation coefficient between the different members of that 17-member community and dopamine. And so we can see that there are metabolites that are positively correlated with the abundance of dopamine, and other also microbes that are anti-correlated. So very much like what we saw with serotonin. And if you do growth curves on the isolate organisms, you see the same sort of dependence. So like Rhizobacterium, it is positively correlated. And if you look at the isolate, as you add in more dopamine, you get enhanced growth. So that's just exactly what you'd expect. Whereas like this Variovorax, which is also a very important organism in the rhizosphere, it's anti-correlated. And as you add too much of it, the bacteria is inhibited. So we thought it'd be really neat to make a plant mutant and test this. But of course, we had a problem here in that the dopamine pathway isn't well known. Especially this 3-hydroxylation step of tyrosine into tyramine is not well understood. And then there's 2 different possible pathways here. There's this pathway, and then another pathway, which is the dominant pathway in humans. So we didn't know which one this is. So we decided to do a metabolite GWAS study, where we took 100 different accessions, and we did exudate analysis, and then looked for genes that were associated with the abundance of dopamine. And we were able to find that on chromosome 2, there were multiple different genes that were all within the same region that were correlated with the abundance of dopamine. So that was neat. So then, and this took forever, as you can imagine. Dr. Vogel, and then also Yizhang Deng, and Yizhang was the person that led this work, used a CRISPR-Cas9 induced genetic system to engineer plants with alteration in pathways, right? So make pathway mutants. And we're able to confirm specific genes that, when removed, decrease the dopamine. So this is dopamine in the roots and the shoots. And here are different genes of interest. And so like with this PPO gene, when you knock it out, you see no dopamine production. There's also carboxylases. When you remove them, you don't have any dopamine. But you have to knock out multiple genes to see. So there's some functional redundancy here. And like this tyrosine decarboxylase, if you knock it out in the shoots, you don't have any dopamine, but there's some redundancy in the roots. So we learned a little bit about this pathway, which is pretty cool. And so now we can do these studies to see, and this is the sort of preliminary study where we're looking at the role of dopamine in the assembly of microbial communities. And we can see a little bit of clustering between the wild type, which is the yellow, and some of these mutants. A little bit of separation there. And here's some examples of the microbes that are either more or less abundant in our mutant. And so we can see that, in fact, we do get some significant changes in the microbial community structure when we remove dopamine from exudates. So that's nice, you know, starting to get to some of these really key metabolites in structuring microbial community. So with that, I'm going to switch gears and talk about the other reason why the rhizosphere is so important, and that is in global carbon cycling, because I want to highlight some recent work with Daniel and Ming, who are leading this seminar series. So soils have lost about 500 gigatons of CO2 because of the way we manage soil. So the typical way of farming is to go drain a wetland that has a lot of dark, rich soil, right? And then you farm it over and over and over again, or you cut down a forest and burn it, and then you farm it until there's not much soil and much carbon left. And so we have really changed the landscape on Earth to where we have many really eroded and damaged soils. And this is a big problem for humanity. And there's a lot of carbon that's going in to soils, but unfortunately, most of it just goes right back out. So photosynthesis is about 123 gigatons of carbon coming into soils, and 120 of that is immediately respired, like within a year. And so there's only a small amount that stays. So if we want to be able to heal these damaged soils, we need to understand how we might be able to keep more of this carbon in the soils. And so there's this great quote from Hain: "If we feed the soil, it will feed us." And so, you know, are there ways that we can use plants and microbes to try to restore the organics that have been lost from these soils? And there's a lot of reasons why you'd want to increase soil organics. It retains water and nutrients, it promotes microbial diversity, climate resilience. It's exact, you know, when you go look at rich soil, that brown stuff, everybody knows that that's good, right? If you grab a soil and it's yellow and orange, nothing's going to grow in that, right? So how can you convert a soil that is mostly mineral into something that's going to support growth? And so we need to identify the factors that govern carbon residence time in soil. And there's many theories. Historically, it was thought that you get this ex situ polymerization of compounds into humics. Now, this is the traditional view. It's a polymerization reaction. This very influential paper in 2011 was saying that, no, it's actually small molecules, like metabolites that make up soil organics. And then why do some things stay and not others? What governs the residence time of soils? This is super debated still, whether it has to do with absorption processes or the stability of the compounds themselves. And one other factor that is kind of bizarre, but really important to be aware of, is that growing plants, while you might think that it would lead to more carbon in soils, it could have the opposite effect. There's this, what's known as the priming effect, so that the plants release labile carbon, like glucose, you know, I showed you sucrose, that can actually give microbes energy to burn off some of the more resistant carbon or trapped carbon. And so here's a quote. Some estimates suggest that the rhizosphere processes at the global scale may release 3 to 10 times more carbon than fossil fuel burning. So this is a very important effect to study too. So in the ocean, which Daniel's an expert about, it's pretty well accepted that there's some carbon that lasts for thousands of years in the soil, multiple cycling of the ocean. And this is because of the chemistry, the actual chemical structure of the carbon. In the soil, there's no consensus about this, whether this is an important factor or whether it's not. So the first question we have to ask is, what is the chemical composition of soil? And this is something that people have been studying forever, for a very long time. This is a paper from 1881, looking at the composition of soil organics. Selman Waxman, before he discovered this was streptomycin, one of the antibiotics, and got a Nobel Prize for it. He wrote a book on humus. And then Dirk van Crevelen's famous for looking at ratios of the elemental composition of molecules to classify them. And there's these fantastic instruments like FTICRs that have unbelievable mass resolution, so you can get the chemical formula directly. But one thing that's really important is if you want to link this to biology, understanding the chemical structure is critical because enzymes can discriminate, microbes can discriminate between, say, glucose and galactose. So we need to actually get to chemically resolved molecules. We and a few other labs have been doing soil metabolomics for a long time. And the advantage of metabolomics is that you're doing chromatography, so you can separate what you're separating the molecules. You guys are all familiar with this. And then you can also do tandem spectrometry, which using a variety of tools can give you structural information. And there's a big need to improve our understanding of and our ability to identify metabolites from these environments because they're very different from what's found in humans, for example. And much more diverse. And so where you might be able to identify, say, 10 or 20% of metabolites in the metabolomics experiments with an animal, with an environmental sample, it's much less than that, or at least that's been our experience. So Ben Bowen had this idea that we've been collecting using the same LCMS methods for a long time now, and we have over 300,000 runs. Maybe we could use this to do some sort of global analysis. Right? And maybe we could use chemical networking, like what's available in GNPS2, to link known and unknown molecules together using cosine similarity type metrics to be able to propagate annotations from knowns to some of these unknowns. So this was a big lift in lots of scope creep. We started off, we were just going to do this for soils, and then pretty soon it became, let's just do everything, everything on Earth. And we actually ended up using, Ben and Thomas used Perlmutter, which is the 5th fastest supercomputer on Earth, to do some of these steps to build a chemically defined network. And so in total, we took samples from freshwater, soil, seawater, and some standards, collected over 5 million tandem mass spectrometry spectra. We used mass difference matching, which is something that Daniel and others had developed for being able to deconvolute chimeric spectra. So, the isolation window on a quadrupole is about a Dalton. And so with these really similar molecules that have similar retention time and similar masses, you can actually get capture multiple precursors with your quadrupole, and then you get these mixed fragmentation spectra. And so this is a really powerful approach for deconvoluting them using some knowledge of the chemistry of these systems. And from this, we were able to see some cool things, that there's a lot of shared features across all of these different environments. So this gives you this impression that maybe there is some common processes that are happening in all of these different environments. There's also some unique molecules that are found in soils and seawater and freshwater. And so just as I was saying, kind of our hypothesis that if you have an unannotated network and you were able to match some standards, you can go look for where you have unknowns that are adjacent to two known identified matches based on authentic standards, and so you can propagate the annotation to this one. When you do this, you go from a 4% annotation rate to a 38% annotation rate. Of course, you don't know exactly which molecule it is, but you know it's very close. And so this is the environmental molecular network where it's colored by the different chemical classes. We are just doing C18 negative. So these are hydrophobic, negatively charged molecules. So this is a thin slice of what's there, but you can see the diversity of compounds that were detected. And this is looking at clustering of the samples. And so up here we have tropical soil, and then we have desert soil, vegetable garden soil, agricultural soil. This is the seawater. Permafrost is up here. And so we can see some, the biggest separations between the marine samples, water samples, and the soil samples, some clustering. So it kind of behaves the way that we would expect. And so we had this fun idea to come back to this idea of, does the structure of molecules actually have anything to do with their persistence? And so we designed this experiment. This is the work of Marcus de Rod, where we took soil, we extracted the organic matter with water. So this is dissolved organic matter. And then we incubated it with microbes from the soil too. So we extracted the microbes and we added them. And we also wanted to look at this priming effect. So we added labile metabolites in one of these conditions to the soil organic matter. And so this is sort of ideal conditions for microbes to degrade carbon. There's no minerals, there's nothing there to protect it. And then the idea was we could do the metabolomic analysis using the environmental molecular network, and we could identify things that don't change, right? And as well as the things that do change. And so this would give us some idea of which compounds may be more recalcitrant to degradation. And so this is the growth curves of the microbes on under these different conditions. And you can see that when we add the labile carbon, we get more growth, exactly what you'd expect. But, and this is with a high level of the labile carbon. With lower levels of the labile carbon or no labile carbon, we get pretty comparable growth. So the microbes are using the organics that are present in the extract. And then we constructed these volcano plots. And so this is looking at the fold change on the x-axis, the log2 fold change. So the decreases, increases, and then we have the log 10 of the negative log 10 of the p-value. So anything above this line is significant. So these are the metabolites that we call preferred. They were used by the microbes. These are the ones that are produced by the microbes, and these are unchanged. So then we had this idea, Ben and Thomas Harwood had this idea of being able to train a machine learning classifier to predict whether a given spectrum will be preferred or produced or ignored. And so they did this using a random forest classifier where they took the data and they split it into a training and a testing set. And then we added the metabolites that they had predicted as being persistent or labile. We added them to soil, we incubated them for a week, we did the targeted metabolomic analysis. And we found that we had all but 2 of our predictions were correct. So this works pretty well, where we were able to predict whether a metabolite is going to be persistent or labile. The ones that we got were incorrect were our tessinate and this prostaglandin. And we think that's probably because these are actually really quickly converted into other molecules. That's what's happening there. And so we think this just shows some initial evidence that chemical structure really does matter everywhere, not just in the ocean, but also in soils. But obviously there's a lot of other factors in soils. So we want to accelerate these types of experiments, do more and more of them to study the role of metabolites in the rhizosphere, both for promoting plant health, but also for controlling this carbon cycle. And so we built this thing called the EcoVibe. And so it has a growth chamber with all these little ecofabs with the plants. We have a built-in microscope, hyperspectral imaging system. There's a robotic arm that, so each of these little, ecosystems, these ecofabs, has a barcode, and this robotic arm gets the ecofabs off of the deck, changes the media, collects samples for metabolomics, images where the microbes are located, the root morphology, and then the hyperspectral camera tells us about the health of the plant. And we are looking at 3 different views, so the top view, the side, and then 2 different side views. So then you can estimate the size of the plant. And since you can do the imaging non-invasively, you can look at how the microbes are affecting the growth rate of the plant over time. And then we've had the great pleasure of working with people that are part of this Camera Institute to adapt the system into an autonomous experimental system. We're still working on this, but as I mentioned, Danny Ushizima and then also Peter Zwart have developed software for automatically extracting the imaging data so that this GPCam software that Marcus Nowak has developed can design the next experiment. So the idea is you feed it an experimental design, and there's some AI tools for designing it, for coming up with the initial design. And then it incorporates the results from your first run to design the next experiment, which then is run by the robot. So I think this is a really exciting future direction to head in. So just to quickly summarize, it's opportunities to use knowledge of rhizosphere processes to select beneficial microbes and potentially manage soil carbon cycling. Fabricated ecosystems provide a path for replicable team science and mechanistic studies. Dopamine and exudates can select for alpha proteobacteria and promote plant growth. And this new environmental molecular network, which is available already as a GNPS2 workflow. So you can use that right now. It enables both compositional and functional analysis of soil organics. And then metabolomic analysis can help us understand the stability of soil organics. And with that, I'd like to thank all of the people that contributed to the work. I see I don't have Thomas Harwood on there, but he definitely did a lot of key work on the environmental molecular network. All of this work was funded by the Department of Energy, and many of the technologies I talked about are available through the JGI user program. So if you're interested, let me know.
Awesome, man. This is so cool.
Yeah, I was a little ambitious with my talk today. I hope I didn't go too fast.
No, this is really good. I'm blown away. I'm sure many people have questions. Let me know if you have any openings at Berkeley Lab soon. But yeah, before we head over to the Q&A, I just want to thank you again, and thanks everybody for stopping by. It's great. We don't have a seminar in 2 weeks because of spring break, but then we will be back in 4 weeks with the VMOL series. So yeah, thanks again, everybody, and good to see you all.